sounds sexier. And you'll meet the rest of the team later. But for now, I would like to share with you my enjoyment in my time in Mexico. I come from Peru, and I have traveled to a lot of places. I've seen a lot of natural wonders, but few times have I been as shocked as when I visited this place, which is Calle del Salto in León. That was 2 days ago. And so that started to make me think about traveling and what are. Does everybody here enjoy traveling? You like traveling?
Yes.
Everybody here? Okay, so I have something to propose to you. What if for today, just for this presentation, you embark on a journey of my own, and I take you to my home country, Peru. Peru. Because in Peru, there are a lot of natural wonders. There are beauties that you have never seen before, or hopefully you have. But there are also problems. And right here, in this green area that you see, that is the jungle, there are enormous problems, especially with mining. And mining occurs in an informal setting where the law cannot reach. So it is totally unregulated mining. And that takes enormous toll not only on nature, not only on fauna, but as well as flora and on people. And so we, these 4 students, take a common interest in that problem, and we are actively trying to solve it. But before you know how we're trying to solve it, you must know who we are.
So, who we are? Well, this group is an iGEM group. So, what is iGEM? Let me explain a little bit about it. Well, iGEM stands for International Genetically Engineered Machine. It is a worldwide synthetic biology competition aimed for undergraduate level students, and it takes place once a year at MIT. The purpose of iGEM is so that groups from all over the world come to find local issues and use synthetic biology as a solution.
For us, iGEM is also a fun way to learn about and engage into synthetic biology. But what is synthetic biology? Synthetic biology is a new area in science that fuses science, math, technology, and engineering. But the high school part of iGEM brings synthetic biology from the world of academia into local community. In this case, our local community, which is Peru. At the beginning of summer, for the iGEM competition, we will be receiving a biological kit, which will have biological parts that we will be using to create our biological systems and operate them in living cells.
So, what does iGEM have to do with GIN? Well, both look at local issues that have a global significance. And in our case, it was water deficits. More specifically, mercury pollution.
3 examples of water deficits can be deficits from human waste, industry waste, and agriculture pollution. Some facts about water. Did you know that the total volume of available water in the earth is 1.4 billion kilometers, and about 70% of the earth is covered in water? But there's a problem. Only 2.5% is fresh water. And even though 2.5% is fresh water, still 700 million people drink polluted water. And also 70% of the industrial waste is dumped into water bodies, which can then be contaminated by biomagnification, which is another process. Now we would like to show you a video about a movie that was made in the Amazonas of Peru, and it talks about illegal mining.
It's almost like looking at the Grand Canyon in America, but then when you actually start to really understand what you're looking at, it hits you even harder. What's happened here, what's happening here.
It's like the wildlands. It's mud and pigs and people. Like I said, when I lived here, the Pituay River was as wide as this sluice area. water was clear, and there was jungle all around. Now it's just rocks.
This is a place where you have basically the highest numbers of any species you can think of. This is Noah's Ark. In this solution, most of the income for this country is based on extraction of natural resources.
It literally reminds you of a war zone after a battle.
We are at the edge.
Yes. So this devastating, both amazing and devastating landscapes are a product of a cycle called Mercury cycle. The Mercury cycle starts with deforestation, which is when all the illegal or informal miners come to the jungle and they log everything out. Then extraction. Then they extract the gold using mercury to purify this substance. And the final step is mercury pollution. When they are done with the gold extraction, they throw all the mercury in the water, and that's where all the pollution comes from.
Okay, now hopefully you've learned a lot in the video, but if you haven't, then I'm going to explain furthermore what is mercury pollution. But first, I would like to state our goal. So, and this is a very important part, because if you don't understand our goal, then you won't understand further explanation in the presentation. Well, what we'll do is we will engineer E. coli bacteria so they can find mercury in bodies of water. So in essence, we are creating a machine that will detect mercury in any water or body that you wish to find. So why are we doing this? This is a very good question, because there are lots of reasons. Well, first of all, this is not only an issue that occurs in Peru, but in many parts of the world. For example, in Colombia, Venezuela, and some places on northwestern Africa. Mercury pollution is something that not many people are aware of. For example, you can see lots of videos about global warming, land pollution, or pollution in oceans. But you don't see much about mercury pollution, but it is indeed a terrible cause, as it has already destroyed 18,000 hectares in the Amazon, and about, in a small survey in Puerto Maldonado, about 60% of adults had toxic metals in their bodies, and 80% of fish had toxic metals in their bodies. And as my peers have stated earlier, the cause for mercury pollution is mainly because of illegal miners. And in the process, they throw their receipts into the rivers, and in the rivers, mercury passes through primary consumers to secondary consumers, all the way up to us. And when it reaches us, lots of terrible things start happening. So some of the effects of mercury in their health are, well, first of all, there's the Acortina disease, which is a terrible disease which damages your kidney, and that doesn't allow your body to clean your blood, and later on you die because you don't have clean blood. And also another disease is Minamata disease, which starts by affecting your neurological system and therefore affecting your 5 senses until you become completely paralyzed and finally you die. So it's some terrible disease. So what have we done until now? Well, we haven't yet started engineering the bacteria, but we have spread the word of what we're doing. We have created a Facebook page, a YouTube channel, Twitter channel. We have a weekly page, and we also have a Fundly where you can donate.
What is IGEM? IGEM is
pioneering ingenuity,
creativity,
innovation, international genetically engineered machine.
What?
ASFM is a worldwide synthetic biology competition that was initially aimed at the undergraduate level, but has since expanded to include high school students and entrepreneurs.
We're proud to say we're the first high school from South America.
IGEM and FPRs are self-dedicated for the Marion High School students for this competition.
We plan on engineering E. coli bacteria in a novel way. We want to address a local issue of global significance and use synthetic biology as a solution. We are working on Project Quicksilver, which addresses the problem of mercury pollution. E. coli that detects and removes mercury.
We're fundraising money to present our work in a GIN conference in Mexico and then in the official IGEM conference in MIT.
And we can't do this without you.
Come on, help us. Help us be a part of the change. Visit our website where you can find more information on how you can help us.
That's the video that we showed.
That's the video that we showed.
Yeah, you can.
So this is a screenshot of our funding page, and as you can see, we have until now raised $800. Our goal is $2,000, but we're not yet there. So little by little, there are many people who have donated, and we need this money so that we can buy the parts of DNA and machinery, so we can start doing our project.
Okay, so our future plans. Our first goal is to educate our community. The reason why is that there are several stigmas and misconceptions about genetic engineering, which make some people be fearful of this technique that we're trying to apply. Have you ever heard that genetic engineering is not good because it is sort of playing God? Have you heard like that? Yes, that's a common misconception. There's a lot of debate around it. And so one of the things that we want to do is bring experts in the field to our school and then educate our community on what we're doing, because what we're doing is safe, and eliminate all that controversy around it. So what would happen if I came here and I told you that right here I had a handful of bacteria that I would throw against you, and then they could easily penetrate your body? What would you do? Come on, tell me. Would you run for your car keys and then leave school?
Right.
No. Would you? Well, some people would. And the reason why is because we perceive bacteria as being something that is always harmful. Well, in reality, you have bacteria in your gut, in your mouth, in your mouth, around us, in the air, everywhere. And some bacteria can be harmful indeed, but some of them can actually help us, just like the one we're trying to engineer. And so, before we continue, I want to ask you, who is afraid of this diagram? Take a look. Raise your hands.
Don't see.
Come on, tell me. I need to know. Who's afraid of this diagram?
Yeah.
You can't put it in your hands.
Yeah. I'm glad. Yeah, I'm afraid of that. It's too weird.
Okay. This is actually quite simpler than what you see at first sight. As I may remind you, what we're trying to do is to get a bacteria to detect mercury. And how we're going to do it is through its genes, its genetic composition. And so, when you want to improve a car, the thing you have to do is take that piece that you want to improve it or change it, and then put it back in the car, and then the car will be better. That is sort of one thing that we want to do. We want to take and extract the DNA of bacteria, improve it, change it, and then put it back in. And this is the parts that we want to improve. And so, is everybody here comfortable with DNA and how DNA works? Okay, so just in case, I'll remind you that DNA is composed of genes. And one gene codes for one protein, and a protein is an agent of change. And so each strand that you see here is a strand of DNA. And all these weird letters, complicated terms, are all genes. The first one that you see, the MER, which stands for mercury regulation. And what is going to happen is that you have an enzyme, which is sort of another agent that will read this chain of genes, and then produce the proteins that are coded for within that. The easiest way to understand this is with a train. Why? You can picture this trend as a railroad. And we see that the enzyme, imagine the enzyme as being the train. And so the train cannot skip a station. And so the enzyme will come and will go in order from MER to promoter operator to MRT, MRP, and then finally to GFP. Just like a train, we have to go to station A, station B, station C, and before reaching station D. And so, what will happen in the bacteria when there is no mercury in the water? This gene will be active, and what this gene will do will produce a protein that will attach here. Who can tell me what will happen if we put an enormous hill in the middle of a railroad track? Will the train be able to go all the way, or will it be stuck? No, obviously, and that is exactly what is happening on in here. When there is no mercury, this gene is active. There's a bump that is produced here, and so the enzyme cannot go all the way. But if you look at the end, you see GFP. I swear I will give the prize to whoever knows what GFP is. Does anybody know? You know?
I knew.
GFP stands for green fluorescent protein. It's something that makes something glow. So if I have a bacteria with GFP, and GFP is being produced, is active, the proteins, the green fluorescent protein is active, is something, you will literally see it glow in the dark. And so what we're doing is we're adding the green fluorescent protein at the end. Picture this as if it was the last station. And what we're caring for is that last station, because we know that if it has reached GFP, and if we know that if the bacteria is glowing, then the train has gone through station A, station B, station C, and station D before reaching its final destination. And so as I told you before, when there is no mercury in the bacteria, literally no mercury, MER will produce a bump in the entire place. And so it will not glow because the train will not arrive at GFP. But when there is mercury, that bump is removed because the mercury, as you can see here, attaches to that protein and takes it out. And so this enzyme is now able to go all the way. So it will produce protein coded for in MERRG, skip this region, MERT, MERP, and then final GFP. And these three must be produced together, just like a train cannot skip a station. And so we know that when we see the bacteria glowing, that all the other genes have been read. Just like when we see the train at the final station, we know that for a fact the train must have been through all the other previous stations.
Alright, so now we're going to guide you through the process of how we're actually going to assemble the genes into the bacteria. And since you saw that we're going to have to create our own genes from scratch, well, so are you. So we have a little activity for you. And you have to split up into groups of 3.
Es única interactive.
Sí, esa es una actividad.
Pero tienen otra.
¿Lo puedo sacar o no?
¿Tienen otra actividad?
Te voy a preguntar, ¿lo puedo sacar o no?
Déjalo en la bolsa.
Oh, okay. I just like
Okay, so if you look at the instructions, you're going to see a part where it says the gene. And you're going to see a lot of beads that are not put together yet in the little bag. So you're going to have to actually build the gene. And we're going to be in the room available for any questions or help if you need to. So that's what you have to do right now.
Is it like time? No. Okay. Si the other one can go together, I will go.
So,
¿El pelado? Pelado aquí es diferente a pelado.
vamos contra ustedes el lunes. Tenemos juego el lunes contra ustedes. ¿Sí? Ya valieron.
¿Dónde es?
¿Qué?
¿Dónde es?
A las 6 en su cancha y siempre perdemos el día.
Si es aquí, si es en las del
Ya, este es el gene.
Venga, yo digo.
We're trying to clone. Pero todavía no.
¿Y esto qué es? Esto ya está.
This is the gene that we're trying to create. This is the gene that you're going to cut.
Pero el cuerpo no tiene...
You're going to create this gene. You're gonna put it in here by cutting.
¿Cuánto crees que se tardan?
¿Qué?
¿Cuánto crees que se tardan?
En hacerlo hasta July.
Así le van a empezar.
July
Se
lo
tienen que
comenzamos.
inyectar y desinyectar para poner esta bacteria.
No.
¿Cómo la haces?
¿Cómo nos tiran la bacteria?
Sí.
¿Cómo la agarramos?
¿Cómo la meten?
Because we received from MIT, como te dije que íbamos a recibir un biological kit y con ese kit ahí tenemos toda la bacteria y todo. Y ahí hacemos el genetic engineering to create this.
Pero,
That's gonna glow on Mercury.
¿cómo se la metes a alguien?
No se la metes a alguien.
No.
Se la metes a un...
¿Cuánta Mercury en el mundo?
Se la metes a la bacteria.
Sí. Y pues esa... Pero,
Y la bacteria no se la metes a nadie. La pones en agua y if the water is contaminated, it glows.
yo pensé que para ver si gente tenía mercury.
Pero ¿cómo le vas a hacer para...?
No podemos hacer eso.
Pero, ¿qué tanta Mercury hay en el mundo?
En el mundo no sé cantidad, pero en Perú el legal mining is the main problem. O sea, 75. Sí.
En Monterrey
If
nos pasó nada.
you lived if you were to live in the jungles of Peru, you will be contaminated with mercury. Around 75% of people there are contaminated with mercury, and they have diseases like they cannot move, they become paralyzed. Pero también, Perú es number one.
Esta bacteria la van a tener que en un solvent.
¿Quién entiende?
Como 75% de la población del mundo.
No, 75% of the people in Puerto Maldonado.
Por eso en Perú.
Sí.
Y ya que la bacteria la van a tener líquido, ¿ok? O sea, se la van a echar al...
Delgado, imagínate, you have a glass of water, and you put one droplet of that glass of water into our bacteria. If it glows, it means the mercury. And you
el
cannot
agua
drink
a la bacteria.
it. Claro. If it glows, you cannot drink it.
¿Y cómo van a producir suficiente como para que todo el agua del mundo...
Once the first bacteria is produced, then it starts producing alone.
Se dividen solas.
Se van dividiendo until we have all the current one.
Esto va a ser, o les va a ir con madre o les va a ir un jefe. ¿Qué? Sí, porque se ve demasiado fácil para que sea verdad. O sea, que pierda. ¿No crees que lo vayan a hacer? Yo creo que sí, porque ese güey sí se ve que sabe lo que está hablando.
Tiene que crear eso.
Yeah, no, I can see how that's, I mean, that's, what they're doing there is just devastating. I'm gonna go see the film, but it's really impactado. I was in Peru recently traveling and,
If you go by plane to Brazil, you literally see.
That's really sad, but one of those issues is the same with the elephants in Africa. And all these things you get that there's people in poverty which don't find other alternatives, so they do illegal things, and it's hard to push them away from it.
I think that the government should not penalize, but incentivize them
To do something.
They want
The penalization is always, it's just pointless.
Ok, so ahora aquí va.
Guillermo. Ya terminamos.
No, hay que ahora poner todo hierro.
Sorry.
What? No, man. Tiene que ir aquí, ¿no? What? Tiene que ir aquí. Why? Digo, está más chiquito.
No, that's weird.
No.
Look. This is what? It's like what? It can be together.
You're the teacher.
Here. It has to be. It has to be here. Look. Yeah, that's right. That's right. Yeah.
Are you all guys fine? Is it
Yeah.
going well?
Es un maestro.
Master?
Es un maestro.
Ah, sí.
Es de Australia.
No, es
Okay, so
Reino Unido, o sea, nació en...
if you want to do something, go. Like, go.
Yo tengo aquí escrito.
Here's a cut.
¿Ya?
Y él también, él es el maestro de biology en su...
No, él. El profesor de biología no puede venir.
No.
Sí, exacto. La diferencia es que los palitos siempre van en la dirección pero sí, mi cuerpo se los expliqué.
Okay, so... ¿Qué horas?
Sí, Nina.
Yeah, you guys are cutting it.
Nina.
Jeans.
Okay, everyone, are you finished? Yes. Yes? Okay, so now I'm going to guide you through the steps to actually insert the gene into the bacteria. So this process is called the 3ASFM. Okay, so step 1. We have our bacteria, E. coli, and we have two different parts that we want together. And so first let me explain, that little ring of DNA that you have is called a plasmid. And so the plasmid is divided into two parts. First, there's the gene that you want, that would be part A or part B. And then everything else that you don't want is the backbone. So the backbone that we're going to be using is that one. I don't expect you to memorize the name, but next. Then we have to actually take out the plasmid from the bacteria. So we kill them. They pretty much blow up and we get the plasmid from there. And we have it on its own. And now we have to actually cut the part out of the plasmid to remove it from the backbone. So you're not actually going to have to put two pieces together, but you are going to have to cut your plasmid to put this piece into it. So next. And the way we're going to do this is with restriction enzymes. And so what these do is when they read DNA, they run into a pattern and they cut it. Just like this. And these are the four that we're going to be using. So what we end up with is these things called sticky ends. Because if you see the pairing in your sheet, these bases always stick to each other. So the enzyme you're going to use is XPAL, the first one. So we gave each letter a color that matches your genes. And you're going to find these patterns to cut it in your ring.
Okay, you do what? Red, green, yellow, green.
Intenta encontrar este patrón de colores.
¿Cómo lo voy a cortar?
¿Están entendiendo? Sí. No, dime si no.
Yo no entiendo, tiene que ver con que cada...
No, esto, la presentación.
Ah, sí.
Tienes
Un rojo.
Okay. Y el de abajo.
¿Quién lo hizo? ¿Quién hizo más trabajo? Es E. coli.
¿Y qué haces también?
Yo pensé en inglés. Para practicar.
¿Todo bien?
claro, o sea...
Sí, pero
no. Entonces, si es que estos son los colores, acá es donde tienes que cortar. Entonces,
No, it's green.
Gratis no.
No, ustedes van a ver dónde están de votar.
Ah, sí.
No. Been having trouble with somebody assembling it incorrectly.
You.
That was not me. Incorrect.
Solamente con el yellow, the green
Ya nos quedamos sin tiempo.
Tienen 9 minutos.
Sí, vamos. Ya, Nina.
Ya, entonces, este...
Okay, so now we have all our parts aside from the backbones, and then now we have a problem, and that's that if we have the backbones and the parts together, naturally they're going to find each other again and stick to each other, and we don't want that. So we add a different backbone to it, that's the same, and what makes this special is that it can hold both parts together. So when we combine all of these things together, then you're going to end up with 3 different outcomes. You're going to have some backbones that are going to have only part A, some are going to have only part B, and then some are going to have both parts together. So now, you're going to actually put the gene into the plasmid. So like, remember how we had the sticky ends? You're going to combine it with the plasmid. How does like now? Yeah. Okay. All right, so the next step is we have... okay, so we have the 3 different options, and we put them back into the bacteria. And so we're going to have 2 bacteria that look exactly like it did before, and then the one that we want that has both parts. And next. So something else that's special about the new backbone is that it has resistance to chloramphenicol. And so when we expose the bacteria to this chemical, then the 2 original ones are going to die. And the one that we want is going to live and procreate. So we're going to end up with a culture of bacteria with both parts. Okay, next.
And so, for the group that completed the activity fastest, we have a small prize, which is actually E. coli, bacteria, little, I don't know what these are. So, these are for you. I don't know. And so I would like to mention the IGEN conference, which is the largest synthetic biology conference anywhere in the world. And that is what we're aiming for, and that will take place in October. And I'm personally excited because what we're addressing is a local problem of global relevance. Informal mining takes place in Peru, but also happens in other I would even go as far as to say that it happens in most, if not all, other sub- or underdeveloped countries that have a potential for mining, as well as some other developed countries. And so when we reach the IGEN conference, we'll find people who are like-minded and are working on similar projects.
And also, before I said, what can you do, I would like to say, what are we going to do next? As Ryan well said before, all great projects start small and take a lot of time. Our project is going to start small, but it's going to take a lot of time. But when we achieve our final goal, it's going to have an impact locally and globally on our communities. We've already made some contacts with doctors in Caetano area, which is the main university of medicine in Peru, and we're planning to proceed with them to make new projects. What can we do? Well, we have social medias like Facebook, Twitter, we have a Weebly page, and we also have a Fundly page, and you can donate, and if you're not ready yet to donate, you can become a supporter, and we will really love it. And finally, with all this information, how can you start your own lab? Well, you can visit igem.com, and they're going to have instructions to how you can apply your learning that you learned today into a new genetic engineering program.
Okay, so before we end, we'd like to thank our main sponsors, Michelle Yuhas, who donated $100, and Sarah DuPont, who is also the producer of the film Amazon Gold, the one we showed earlier, who donated $200.
And we have time for 2 questions, if you have any.
How did you come up with this idea?
That's a good question. And I feel like in Peru, this is probably the biggest problem that the government is facing. And the media surrounds us with all these types and all these pictures of the destruction in the Amazon and the people who are being poisoned. And I personally fear that Lima, being in the 2nd largest desert city in the world, is going to quickly run out of water from the glaciers in all the water that comes from the melting glaciers in the highlands. And so I think that we should start taking a greater care of the water. So yeah, a lot of factors that is what we ended in.
You guys mentioned something about silver, I don't know, equipment, yes? Or was it just silver fish?
Silver, what do you mean by silver?
You said something about a silver equipment.
You mean a silver sponsor? Or?
Well, another question. Like, what is the equipment that you will use to make these, to analyze all the bacteria to your experiments?
So how we're going to get it, or
what's the machine? Like, yes, what's like...
You're asking about what we need to use. Okay. We have a long response to that short question. And that is, we need first a machine that can create and amplify and generate all the genes that we want. And so as you saw that chain of genes that I showed you in my slide, we need one machine that is capable of creating those, a lot of billions, 30 billion copies in 8 hours. And so then we need a machine to test whether these genes are correct, and then a minus 80-degree freezer to keep the cells in. And yes, we're slowly acquiring all these machinery. And that Fundly page that you saw at the beginning, that screenshot, all that money that is being raised through that avenue is going directly to purchasing the machinery. Because the method actually is complex, and the machinery is complex as well, but yes, we have it.
Yes, alright. I'm guessing, well, you guys didn't mention it in the presentation, but once you finish with the bacteria and everything is done, what do you plan to do with it? Of course, you guys try to implement it, but what are you going to work with the government or create your own business or what?
Yes, great question. We've already made contact with a doctor, with the head doctor in Cayetano Heredia University, and we've already made some plans, and we plan to create a kit with our bacteria that will go to the poor communities and to the local communities that are suffering this mercury pollution problem, and we're going to take that kit to them, we're going to give it to them, and that way they can put the water they're supposed to or they're going to drink into that kit. And if the bacteria close, means that the water they're trying to drink is contaminated. And that way we prevent that they drink contaminated water.
Yeah, we aim at doing something similar to what airport security does in the airport. I don't know if you've ever been fortunate enough to see this, but when the security in the airport fear or suspect that a substance such as a water bottle contains cocaine, what they do is they have a small sachet which they open and which contains liquid that will turn blue if cocaine is present. Similarly, we want to have a small sachet where the people can test a single drop of water, and if that water, which is extracted from the main water source, such as the river or the community pool, whatever it works, then that sachet will turn blue. And then you'll immediately know that the water is contaminated and is not safe to drink, therefore saving several lives and costs, improving the quality of life.
Also, like, the current method to detect mercury in water is with a mass spectrometer, which is very expensive. So, like, I can guarantee no one's going to use a mass spectrometer in the middle of the jungle. So, we'll make it more accessible to people who actually need it.
Okay, guys, so
Do you have any questions or?
You can ask your friends if you have any more questions. Now we're moving on to dinner.
Yes, to
the cafeteria.
Hey guys, one last thing before you guys leave. iGEMS is something that you can start in your own school as well. So if you're interested in doing something similar, talk to these guys about how you can start doing some type of genetic engineering at your own school.
Okay.